Our featured presentation is an extract from the book, Diary of a Fat Enzyme. It's portrayed in the form of a video to enhance and grasp the attention of all audiences. Enjoy.
Tubby's dropped off by his mom 10 minutes late to school. Tubby hears his name over the PA system and quickly reports to the nurse's office.
Tubby dear, I'm sending this note home for your mummy. Make sure she gets it. It is very important.
Mummy, here's a note from the nurse of my school.
What does it say, dear?
I don't know, but I think I need to go to the doctor for a checkup.
Well, that's okay. Your dad and I will take you at once.
Good day, my boy. What brings a fine young man like you to my office today?
His school nurse recommended that he be taken to see a doctor, in case of any illness due to his weight.
Well, I can put my knowledge of his BMI against that of the expected BMI for young boys his age.
Whatever is necessary, doctor.
Meanwhile, Carl and David are engrossed in a conversation at work when Carl suddenly speeds off to the bathroom.
Hey David. I'm so sick this morning.
How come? How are you feeling?
Oh no. I have to go to the bathroom.
Hey David, have you seen Carl for the morning?
Here he comes.
Oh dear.
While walking up to David and Laura, Carl blacks out and falls to the floor. He is then rushed to the hospital. Around the block from work, his colleagues along with his wife, wait in anticipation at the hospital to be briefed by Dr. Jason Matthews.
Good day, I'm Dr. Matthews. Carl has retained consciousness and is stabilized. We're about to conduct a few tests to see what may have caused his episode today, after which he is free to leave, and as soon as we have a proper diagnosis we'd contact him.
At this time Dr. Frank gives insight into Tubby's condition based on the tests he conducted.
A BMI, body mass index value, is a measurement that has replaced weight, and is the preferred determinant of obesity. Tubby's BMI was calculated to be 32. This is a clear indication that Tubby is indeed overweight, but more so that he is obese, as obesity is characterized by a BMI of 30 and over. Obesity is not a disease in itself. It is a condition that increases a person's risk of developing health problems such as type 2 diabetes, heart attack, stroke, breathing problems and some types of cancer. Tubby's test results reveal that his weight has caused him to develop type 2 diabetes. Type 2 diabetes, or non-insulin dependent diabetes, is a metabolic disorder characterized by an increased blood glucose level coupled with insulin resistance. It can be initially managed by exercise and dietary modification. However, if that fails, then insulin can be prescribed.
But what exactly causes type 2 diabetes, doctor, and what are some common symptoms?
Some common symptoms include frequent urination and hunger, weight loss, blurred vision and fatigue. But long-term complications are far worse. So it was within Tubby's best interest that his school nurse asked that he be carried to see a doctor. Long-term complications include heart disease, stroke, blindness, kidney failure and poor circulation leading to amputation. Also, his lifespan can be shortened by 10 years.
Carl had been discharged, and returned to work 3 days later. Carl received a phone call from his doctor telling him that his diagnosis came back positive for type 1 diabetes.
Excuse me for a moment.
Good day, Carl, this is Dr. Matthews. Your ketoacidosis test indicated that you in fact have insulin-dependent diabetes. This is responsible for your frequent use of the bathroom, increased hunger, fatigue, vomiting, dehydration and abdominal pains are also characteristic, so beware. This type is caused by the autoimmune destruction of insulin produced by the beta cells of the pancreas located in the islets of Langerhans. Insulin is a peptide hormone required to decrease blood glucose levels. Both you and your wife are invited to a seminar hosted by a group of esteemed doctors who wrote the book Diary of a Fat Enzyme. I am positive that you'd gain a lot of insight into your condition. I hope to see you.
Later that afternoon, Dr. Matthews met with the doctors who wrote the book Diary of a Fat Enzyme. They discussed the underlying principles of their seminar to be held that week.
Good day, we're here today because of the upcoming seminar we have in 2 days, based on our book, Diary of a Fat Enzyme. We need to go over the mechanism of action of insulin and glucagon. Also we're going to review the importance of integration metabolism and the effect of inhibitors on each metabolic pathway. We are all aware that during the fed state, there is a surge in blood glucose levels. Insulin is an anabolic hormone. It causes the cells to take up excess glucose, particularly important in maintaining appropriate blood glucose levels. Active transport of glucose across the plasma membrane of cells in adipose and muscle tissues is necessary since glucose molecules are too large to simply diffuse across the membrane. Glucose transport protein GLUT4 is the transporter of glucose and is stimulated by insulin. Firstly, the insulin must bind to the insulin receptors located on the plasma membrane of the cells in the targeted tissues. The insulin receptor is a transmembrane receptor and belongs to a class of tyrosine kinase receptors. It is a tetramer composed of 2 extracellular, alpha subunits and 2 beta transmembrane proteins. Since insulin is a polypeptide hormone, it is too large to cross the membrane, and so it binds to the alpha subunit. As insulin binds, the receptor changes its conformation. A tyrosine kinase enzyme located on the beta subunit is activated and autophosphorylates tyrosine residues also located on the beta subunit. Tyrosine kinase also phosphorylates tyrosine residues on a protein called insulin receptor substrate 1 or IS1. The activation of this protein causes the activation of several signaling pathways that control a diverse array of effects. The result is a phosphorylation cascade, which is the phosphorylation of key enzymes. Insulin activates protein phosphatases and dephosphorylates these enzymes. Some of these are activated by phosphorylation, others are inactivated through the same mechanism. Insulin activates glycogen synthetase and pyruvate dehydrogenase and inactivates phosphofructokinase 2 and hormone-sensitive lipase. Another effect is the activation of phosphatidylinosytol 3 kinase, that fastens the translocation of GLUT4 containing vesicles to the cell surface. This is important to allow uptake of glucose by skeletal and fat cells. Now, as insulin binds to the receptor, the GLUT4 transporters, located in intracellular vesicles, are quickly translocated to the plasma membrane and increase glucose uptake from the blood into the cell. When insulin levels decrease, the GLUT4 transporters leave the plasma membrane and return to their intracellular vesicles. Glucagon is the main hormone of the fasting state. The blood glucose levels begin to drop several hours after a meal, leading to a decrease in insulin secretion and a rise in glucagon secretion. Glucagon is secreted by the alpha cells of the pancreas in response to a low blood sugar level. The main target organ of glucagon is the liver. Like insulin, glucagon is a polypeptide hormone, hence it also needs a receptor to bind to. These glucagon receptors are located on the plasma membrane of the liver cells. The receptor is a G-protein coupled receptor, which means that there is G-protein attached to the receptor. G-proteins are a family of signaling proteins. The G-protein is composed of alpha, beta and gamma subunits. A GDP molecule is attached to the alpha subunit. When the glucagon binds to the receptor, this GDP molecule is replaced by a GTP molecule, activating the alpha subunit. Upon activation, the alpha subunit dissociates from the receptor and attaches to the enzyme adenylyl cyclase, activating it, using ATP cyclic adenosine monophosphate, or CAMP, acting as a secondary messenger binding to an enzyme called CAMP-dependent protein kinase or protein kinase A, activating it. Protein kinase A leads to a wave of phosphorylation of the key enzymes in the metabolic pathways. This enzyme activates phosphorylase kinase, which, in turn, phosphorylates glycogen phosphorylase, converting into the active form called phosphorylase A, which is the enzyme responsible for the breakdown of glycogen.
The body's metabolism consists of many pathways, which are highly interconnected, and perform their respective functions simultaneously. Each major metabolic pathway has an intricate network of reactions, which are able to sense the status of their coexisting partners, to ensure that they are functioning optimally. Integration of metabolism is thereby important, as it is responsible for the regulation and proper functioning of the body with respect to the generation of ATP for energy. The processes involved in the major metabolic pathways are regulated to ensure that reciprocal processes do not occur at the same time. There are 3 crossroads in regard to the flow of products of the different metabolic pathways. These key junction points are glucose 6-phosphate, pyruvate and acetyl-CoA. Therefore, integration metabolism is essential on both a long and short-term basis for the maintenance of the major metabolic fuels, glucose, amino acids and fatty acids. Glycolysis and gluconeogenesis are coordinated, so that within a cell, one pathway is relatively inactive, while the other is highly active. In glycolysis, phosphofructokinase 1, or PFK1, is the key regulatory enzyme that catalyzes the irreversible transfer of a phosphate from ATP to fructose 6-phosphate, giving fructose 1,6-bisphosphate, and ADP. When ATP levels are high, the cell no longer needs metabolic energy production to occur. Thus, PFK1's activity is allosterically inhibited by ATP, and high levels of citrate, another high-energy marker of a cell. On the other hand, fructose 1,6-bisphosphatase is the main regulatory enzyme of gluconeogenesis and is activated by high citrate levels, and inhibited by high levels of AMP. Glucose 6-phosphate dehydrogenase catalyzes the committed step of the pentose phosphate pathway. The enzyme is regulated by the availability of the substrate, NADP+. As the NADPH is utilized in reductive synthetic pathways, the increasing concentration of NADP+ stimulates the pentose phosphate pathway to replenish NADPH. In fatty acid synthesis, palmitoyl-CoA, the product of fatty acid synthase, promotes the inactive conformation of acetyl-CoA carboxylase, diminishing the production of malonyl-CoA, the precursor of fatty acid synthesis, thereby inhibiting the reaction. Finally, the day of the seminar arrived. From the far left was Dr. Fred, Dr. Matthews, Dr. Mitchell, Dr. Findlay and Dr. Frank.
Insulin is secreted in response to increased blood glucose levels during the well-fed state after the ingestion of a meal. Its function is to lower blood glucose concentration by stimulating the removal of excess glucose from the blood and introducing them into cells that require glucose for energy, such as brain cells and erythrocytes. If there is an insufficient supply of insulin being secreted, glucose is ineffectively absorbed into body cells, resulting in the blood glucose level remaining high. This occurs in individuals suffering from diabetes, and requires the introduction of insulin into the body, in order to lower and regulate the blood glucose level. Glucagon works antagonistically with insulin, opposing its effects, and plays a major role in sustaining glucose levels during the fasting state, by stimulating hepatic glucose production. This production of hepatic glucose is primarily regulated by glucagon through the breakdown of glycogen stores into glucose. This maintains blood glucose levels within the normal range when they become significantly low during the fasting state. When the fasting state comes to an end, the ingestion of a meal causes glucagon secretion to be suppressed, resulting in the secretion of insulin once more, beginning its action on the increased blood glucose levels.
Carbohydrates and proteins obtained from the diet in excess are converted to fatty acids in the liver, which are stored as triacylglycerol, TAG, via the reactions of the glycolytic pathway and the citrate acid cycle. The committed step of fatty acid synthesis involves the formation of malonyl-CoA by acetyl-CoA carboxylase. ACC is the main regulatory step in fatty acid synthesis. ACC is a bifunctional enzyme and has both a biotin carboxylase site and a trans-carboxylase site. Each cycle, however, requires the input of one malonyl-CoA and the oxidation of two NADPH2 molecules. Beta oxidation is the major pathway for catabolism of saturated fatty acids in the mitochondrial matrix. After long-chain fatty acids enter a cell, it is converted to its CoA derivative by the enzyme thiokinase. However, the inner membrane is impermeable to CoA. As a result, CPT1 located in the outer membrane replaces CoA with carnitine, forming fatty acylcarnitine, which is translocated across the inner membrane. The carnitine, translocating protein, is an antiporter protein that exchanges the products at each site. Once inside, the fatty acylcarnitine is converted to fatty acyl-CoA by CPT2. Beta oxidation occurs via 4 enzyme-driven steps, each of equal importance. The first step is end oxidation using acyl-CoA dehydrogenase to produce FADH2. Second step involves hydration by use of enol-CoA hydratase. Third step is second oxidation to produce NADH by using 3-hydroxyacyl-CoA dehydrogenase. Final step, ethylytic cleavage to release a molecule of acetyl-CoA. The final cleavage, however, produces 2 molecules of acetyl-CoA. Subsequent oxidation of FADH2, NADH and acetyl-CoA by the citrate cycle, and oxidative phosphorylation, generates large amounts of energy in the form of ATP.
Melanyl-CoA is the 3-carbon derivative of acetyl-CoA, and is the major product of fatty acids, and the intermediate of long-chain lipid synthesis. Melanyl-CoA has 2 important roles in both lipid and carbohydrate metabolism. Its formation can be described as the committed step in fatty acid synthesis. Melanyl-CoA also coordinates the oxidation of fatty acids. Acetyl-CoA carboxylase, the enzyme that catalyzes the formation of melanyl-CoA, is strongly inhibited by phosphorylation by AMPK, also known as AMP kinase. An increased level of AMPK can therefore turn on fatty acid oxidation and mitochondrial synthesis of ATP. The increased fatty acid oxidation would then turn off carbohydrate oxidation. When energy utilization exceeds ATP synthesis, AMP concentration will increase. This in turn initiates the activity of AMP kinase, stops acetyl-CoA carboxylase activity, and reduces melanyl-CoA formation levels. Hence, fatty acid oxidation in the mitochondria is accelerated,